Welcome to our webinar series for professionals.
So, welcome everyone to February's webinar with Dr. Tom Fabian, who has a PhD in molecular biology, and we have had the pleasure of having him teach and do webinars with us before, and it's always exciting, and we learn a lot. But he's also a leading expert in the microbiome in relation to both health, immune function, chronic disease, and aging. So he works for Diagnostic Solutions as a consultant and educator, and that's, of course, what you see him in today. And today's presentation has the title of Advanced Intestinal Barrier Assessment with GI Map. So we are talking leakiness, leaky gut, tight junction proteins, etc. So I'm Anne-Catherine. I'm the co-founder of Nordic Laboratories and DNA Life, where we are, as I say, always dedicated to change healthcare, support healthcare through functional medicine, personalized lifestyle, N equals 1, having the patient person in the center of the treatment, making them be aware of the responsibility that they have in order to be healthy and fit for a long time. So the test that we're talking about today is going to be GI Map, that one, and many other tests, of course, you can find in your practitioner profile on VMS. You also find supplements that you can order there for yourself and for your patients. And reach out to us if you have any questions. Then, of course, we are happy to support you the best we can. Then I just want to say, for those maybe a little bit closer to London, but we do have this in-person event happening in London on the 10th to the 12th of March 2023, where Umaru Cadogan, who we had the pleasure of having fun with last month when he was educating on genetics. And if you haven't seen that, please go into VMS and find it there and watch it. It was a very, very interesting introduction to the ApoE4 gene and the COMT gene. But in London, it's going to be the focus on combining genetics with functional testing. And I just really hope that you will join us. Of course, we've sent out invites already and more will follow. Otherwise, just reach out if you want to do it, or in VMS, there's also a link for education and you can log in and sign up there. So that's enough of my blurb here. So what I'm going to do now, Tom, is hand over the stage to you. Are you ready for that?
I am all set. I'm going to go ahead and share my screen here. Just take me a moment.
Do that, and then I'll mute myself.
All right, so just checking to see if you can see my screen okay.
Absolutely, yes, I can.
Perfect. All right, so welcome everyone. Thank you for joining us today. I'm located in Denver, Colorado. I work with Diagnostic Solutions Laboratory, as Anne-Catherine noted. They're located in Florida, so a little bit of a time difference here. It's early morning here in Denver. So we're going to focus on today is how to assess the intestinal barrier from a more comprehensive standpoint. So this is reflecting a lot of the newer research that has been coming out in the last few years. In our field, there's a lot of focus on leaky gut, of course. That's kind of the top focus when we're thinking of the intestinal barrier and how to assess the intestinal barrier. So, of course, we do have a marker, key marker zonulin, which we'll talk just briefly about at the beginning. That is one of the key markers for the classic understanding of intestinal permeability. But we're going to really take the lens and zoom out today and get a wider picture of the intestinal barrier. So there's a lot more to this. It's a more complicated structure, as you can imagine, than just the tight junctions. So there's a lot of information that we can get from comprehensive stool testing once you know how to interpret the information, once you have the background. So again, that's what we're going to be going through here. So the reason I chose the word advanced intestinal barrier assessment, again, is because we're going beyond just looking at zonulin and leaky gut. But that is a key part of the assessment. So I don't think anyone in this group necessarily needs a reminder of just how important the intestinal barrier is, but this is one of my favorite quotes that kind of summarizes that. The title is Intestinal Barrier, a Fundamental Role in Health and Disease. The gastrointestinal mucosa constitutes a critical barrier where millions of microbes, and in fact, of course, in the colon, it's up to the trillions of microbes and environmental antigens come into close contact with the host immune system. The intestinal barrier defects have been associated with a broad range of diseases, therefore denotes a key therapeutic target. So again, this is really just, I think it bears sort of just reminding every once in a while just how important this is, because this is a critical barrier between our environment and our internal physiology. So there's lots and lots of complexity, lots of interactions. Our intestinal barrier has to perform a lot of functions to both allow nutrients in, but also not allow the things in that we don't want to have and interacting with our immune system, et cetera. So this is really where GI map comes into play, because we get insights into the overall microbial ecosystem, but also the flip side, the other side of the coin, which is the GI physiology. You really need those two components together to put the picture together, because there's so many influences back and forth. And that really allows you to connect the dots. So that's one of the reasons why I like this figure, because it really, when you're looking at GI map, it's about learning the information behind it, so that when you're looking at a patient and their results, you can start to connect those dots and get some great clinical insights. So just a quick review. Again, probably most of you are familiar with the general markers that are on tests like GI map, but let me go ahead and just cover this briefly. And again, many of these do have roles in the intestinal barrier. So we're going to learn a lot about that today. Certainly pathogens are really key, of course, for understanding infections and other processes that may disrupt the barrier, for example, cause inflammation. Similar scenario with opportunistic microbes. Also normal microbes. So we have a great sampling of keystone species and all the way up to the phylum level. So you kind of get the big picture of what's going on with the normal microbes. They play a huge role in promoting a healthy barrier. We'll talk about that. Certainly immune responses, when they're excessive, of course, then that can affect the barrier and vice versa. Digestive function, we're going to talk less about that today, but certainly digestive imbalance is one factor that can lead to downstream issues with the barrier and also with dysbiosis. A range of other markers representing aspects of detoxification, such as beta-glucuronidase, presence of occult blood, even antibiotic resistance genes, particularly for H. pylori, that can help guide treatment. And then, of course, the key marker that a lot of practitioners focus on when they're thinking about how to assess leaky gut, how to assess the barrier, and that's fecal zonulin. So we're going to talk a little bit about that and then move on and kind of zoom out to the rest of these components. And this is a review article from a while ago by one of the key pioneers in defining, identifying zonulin years ago. So it's going back, this review is about 10 years old, but the research goes back at least 20 years. So I'll just read the highlighted part of this research article summary here. It says, our discovery of zonulin, which is the only known at this time anyway, the only known physiological modulator of intercellular tight junctions. So of course, those are the structures that help keep the cells together and prevent molecules from getting through. So basically, it's the only known physiological modulator of these tight junctions. So it essentially regulates them. And that led to increased understanding of the intricate mechanisms that regulate the intestinal epithelial paracellular pathway. So that just refers to when there's permeability that goes through the gaps between the cells. That's the paracellular pathway. Led us to appreciate that it's upregulation. So when it's elevated, especially in genetically susceptible individuals, can lead to autoimmune diseases. So of course, Dr. Pisano primarily studied celiac and related diseases, but now we know intestinal permeability and zonulin are related to a wide range of conditions. Certainly infections, allergies, particularly food allergies and food sensitivities, chronic inflammatory conditions like inflammatory bowel disease, IBS, etc. So very, very important discovery, very important factor to assess. So I kind of want to just start off here and contrast two different patient results just with regard to zonulin, just to kind of lead off this discussion. So just kind of quickly, these are just very short summaries of these patients and their key symptoms and diagnoses. So patient one, diagnosed with diarrhea-dominant IBS-D, acid reflux, migraines, acne, rosacea, Sjogren's disease, as well as mold exposure. And I think many of you already know that a lot of these conditions, issues are often associated with leaky gut, particularly IBS-D, for example. And of course, whenever you have issues in the gut, and then you have systemic issues, so symptoms occurring elsewhere, such as migraines, skin dysfunction, such as acne and rosacea, autoimmune conditions like Sjogren's, certainly we would suspect leaky gut. So we do see that confirmed for this patient here. You can see the zonulin here is all the way up at almost 204, so well above that cutoff level of 175. But on the other hand, we have the second patient here, also diagnosed with IBS-D. Again, we know IBS in general is associated with intestinal barrier dysfunction. This patient also had gastritis, food sensitivities, which are associated with leaky gut, mast cell and histamine symptoms, long COVID, which can affect the gut, and also a history of SIBO. We know SIBO also is associated with leaky gut. And yet at this particular time, when the patient, basically when the sample was run, their zonulin was well within normal ranges, right? So this is a great example of the intestinal barrier, at least in terms of the zonulin marker, certainly is not the only part of the picture that we want to look at. If we're not aware of these other components, we might conclude that this patient doesn't have intestinal barrier problems, but in fact, they probably have pretty significant issues. And when we zoom out on GI map, we can actually see some of those other issues. So again, zonulin is very helpful for many cases, but we don't always see increased intestinal permeability, even in patients sometimes when you suspect it. So that's telling you there may be other issues that are more of a problem for this patient, but also affect the barrier. Let's go ahead and look a little further at this patient's results overall, zoom out, just to give you the bigger picture. So this is the first page of GI map. We can see that for the pathogens, this patient was negative. So they didn't have any current pathogens. We know pathogens can disrupt the barrier. In this case, everything looks good. Same with H. pylori. No H. pylori detected here. So far, no issues that we're seeing. That whole picture changes when we look at the normal commensal bacteria section. So you can see here, there are a couple that are okay. They're trending low at the top, the Bacteroides fragilis, Bifidobacterium, they're kind of trending low, but not officially low. But everything else, every other marker is low here. So this patient is very deficient in their normal microbiome. We know that various microbes play different roles in promoting a healthy barrier. We're actually going to dive into some of that. And that's really kind of getting into the advanced part of the presentation today, particularly Akkermansia, E. coli bacterium, and a few others play especially key roles in a healthy barrier. So you can imagine that when they're deficient, when they're decreased, then that could negatively affect the barrier. On the other hand, under the opportunistic and overgrowth section, we see high Pseudomonas, both at the genus level, meaning that there are additional species beyond just the Pseudomonas aeruginosa. So that particular species is high. That's the best studied of the Pseudomonas species in the gut, but there are others as well that are less well studied. We know those are elevated. We know those are opportunists. Lots of research shows that Pseudomonas can cause a lot of problems in the gut, including potentially inflammation, biofilm formation, may also promote leaky gut as well. We'll talk a little bit about that as well, a little bit further in the discussion here. Under the inflammatory sections, we didn't see anything that was out of range. The first section was the inflammatory and autoimmune related bacteria. These are the more significant opportunists when it comes to inflammation. We generally want to see these below detection limit. We do see that here, so that's good. Then we have the commensal inflammatory and autoimmune species. So these are ones that are always present in most people. They're normal microbes. They do actually play beneficial roles in several ways, but when they're elevated, they can either be a sign of inflammation or autoimmunity, and in some cases, that may actually play a role in promoting inflammation and also autoimmunity. So that's the difference between just the general inflammatory opportunists and then the commensals. Commensals, we do want to see them there because they have some beneficial roles, but when they're high, that can be a problem. So for this patient, we didn't see any imbalances here either. So that part looks good. No fungi or yeast. We certainly know that candida can cause leaky gut. That's well documented. Also can cause intestinal inflammation, etc. So we'll talk briefly about that one a little bit later, but that one was not detected. Some of these other much less common fungi were not detected. Viruses, these are pretty rare. We hardly ever see them. Those were definitely not detected here either, but those also are known to disrupt the barrier when they are elevated. Also no parasites. So we do know some of these parasites can disrupt the barrier, especially blastocystis, dientamoeba, fragilis, etc. Now we get to the intestinal health section. We can see that the digestion markers are starting to trend in the suboptimal range. So steatocrit is a measure of fat malabsorption. Not officially high, but it's trending up into the yellow range. So it's getting into that high normal range. So a little bit of fat malabsorption, not enough to really be considered officially high. Lastase, marker for pancreatic enzyme production, also trending pretty low, not officially low, but this is low enough when it's in the yellow that many patients do have symptoms. And just keep in mind, so we're not going to focus so much on elastase and digestion today, but poor digestion is one of the major causes of dysbiosis. And of course, dysbiosis can cause intestinal barrier dysfunction. So upstream, it is one of the contributors to barrier dysfunction for many patients. And of course, a lot of other symptoms as well. GR markers in this case look pretty good. Beta-glucuronidase was just kind of borderline, not too bad, no occult blood. Secretory IgA, we will be talking a fair amount about that today. That's a key marker for the health of the intestinal barrier. In this case, it was low. We definitely don't want to see that low, and unfortunately, that's pretty common. You'll see that trend pretty consistently with lack of normal bacteria. Not 100% of the time, but you will see that most of the time. And we'll talk about reasons why that's the case. Anti-gliadin looks good in this case, but certainly when that's high, we know that gluten reactivity can cause leaky gut. In general, food sensitivities both can cause leaky gut, but they also can result from leaky gut. So there's kind of that two-way interaction. In this case, that looks good. We will talk more about this. This is one of our newer markers, eosinophil activation protein. So of course, as you can tell by the name, it's telling us that eosinophils are more active than normal when it's high. They play a key role in normal barrier function. So when it's normal, that can be a good sign for some patients. When it's elevated, that can damage the barrier. We'll look at a little bit of the research around that too. This patient did not have that, but just for future reference, high eosinophil activation protein is a significant sign of barrier damage. Calprotectin, of course, I think most of us are familiar with that. Marker for inflammation in the gut, primarily based on neutrophil type inflammation. We all know that inflammation, of course, can cause leaky gut as well. But in this case, this patient's calprotectin looks good. And as we talked about earlier, their zonulin looks good. So that's just kind of reiterating that one looks normal. So we don't want to jump to conclusions that the intestinal barrier is fine just because zonulin is normal. There can be other imbalances. And again, secretory IgA is one of the key ones. So we'll talk more about that. I think it's helpful to kind of when we're talking about all these markers and terms, how do they fit together sort of visually? This is a nice illustration of the barrier, kind of simplistic, but it gives you sort of the key components. So this is kind of the heart of what I wanted to talk about today, is zooming out from the idea of leaky gut and tight junctions, which is this component, right? So it's a little component. It's this structure that kind of ties the cells together. Here are these epithelial cells. And that is an important component when it's when the tight junctions are not functioning well, when there's too much leaky gut, well documented to contribute to diseases and symptoms. But there's also the bacteria. So bacteria not only form part of the barrier because they can repel pathogens, for example, they can metabolize toxins. So they can basically take care of some issues even before they get to the mucus layer or the epithelium. But there's also a lot of interaction. So you think of these components as, at one level, it's easy to think of them as layers with the microbiome, mucus layer, epithelium with the tight junctions, and then the immune system in the lamina propria. But they're highly interactive. So they're kind of functioning as one system. So that's a very key component to understand, is that you need the normal bacteria so that these other components are healthy. That includes the mucus layer, the epithelial layer. So we'll talk a little bit more about that beyond just leaky gut, and especially the key role of stem cells in intestinal regeneration. That's not something that's been focused on a lot in functional medicine, but we definitely want to take that into account. The immune cells that I mentioned that are in the lamina propria primarily. So those are the key components. And just to reiterate, so zooming out beyond just the leaky gut and the tight junctions, we have the microbiome. Then we have secreted factors, particularly mucus. Secretory IgA is produced by immune cells and then secreted out into the mucus layer. So it's a key part of the mucus layer. And then something we won't really talk about today, but also very important, are these AMPs, which is antimicrobial proteins. So there's a range of different proteins that are produced by certain epithelial cells, especially the ones called Paneth cells, that help to counteract pathogens. They have other new functions that have been discovered recently, like promoting epithelial repair. So lots of important functions there. Of course, the all-important epithelial layer itself, with a variety of specialized cells that all have key functions. And then, of course, those tight junctions that connect them together. And then there's the mucosal immune system. So, of course, those are the key components that we want to look at, that we want to assess with GM-MAP. Of course, this is an illustration. Now, those are the key components. This is an illustration of what happens when things go wrong. So you have, at the top, we have basically the barrier damage, and then overgrowth of opportunists, like Staph aureus. Staph aureus is one of the key ones you'll often see elevated, and a pretty long list of conditions, but especially food sensitivities and IBS. Those are two key conditions that are especially linked. And then, of course, other issues related to the barrier, et cetera. But that can lead to translocated microbiome. So you can get microbes in, of course, coming through where they're not supposed to be. That stimulates immune responses, and particularly inflammation. And of course, that leads to, there's a lot of different cells involved here. So when you're talking about inflammation, there can be different types of inflammation. You can have more allergic type inflammation with mast cells and eosinophils. You can have more classic inflammation with neutrophils, and also the T cells and the B cells. So really complicated process, but basically we don't want to get that immune system ramped up because of the defective barrier. So really all this comes down to learning what factors disrupt the barrier. And that's certainly factors that we can see on GM-MAP. And then of course your patient assessment, where you're taking into account diet, et cetera. So lifestyle, diet, stress, all the obvious things. And then factors that promote a healthy barrier. So that would include particularly beneficial microbes. So we are going to talk a lot about that today. But other factors too, we'll touch on a couple dietary supplement factors that are not commonly thought of yet as supplements that may help with healing the gut barrier. So, of course, we typically think of supplements that heal the barrier, such as glutamine, zinc or zinc carnosine, those sorts of supplements. But there's actually much more that can be done to support the barrier. And there are differences, for example, between the small intestine and how you support that, and the large intestine and how you support the large intestine. So it's kind of a complicated topic, as you can imagine, so we don't have time to cover everything today. But certainly, if you have questions about anything in the Q&A, feel free to ask about anything that I haven't covered as well. All right, so let's touch on these factors that promote a healthy barrier. So when it comes to the markers on GMAP, we're mostly talking about the normal microbes. We know certainly diet, healthy lifestyle also promotes a healthy barrier, but normal microbes, very, very important. This is summarized in this review article from 2018, the potential of gut commensals in reinforcing the intestinal barrier function and alleviating inflammation. So you can see here I noted that it's open access, it's free on PubMed Central. So if you want to check that out, it goes through some of the keystone species like Akkermansia and also Faecalibacterium prausnitzii and a few others. This is a figure I use a lot in a lot of my presentations because I think it's just such a nice summary, visual summary that captures those connections between diet and the microbiome, particularly, of course, fiber, polyphenols, healthy components of foods that promote healthy, normal commensals, which then produce short-chain fatty acids and many other products. So short-chain fatty acids are really important, but they're not the only products. But there's really great research, very, very well established, that short-chain fatty acids promote the health of the barrier in many ways, including stimulation of the mucus production, stimulation of secretory IgA. So that's kind of your connection there. When you suspect that the barrier is not really very healthy for a patient, and especially if you see low secretory IgA, you see low levels of species that thrive in the mucus, like Akkermansia, those are all signs that this whole system probably isn't working very well. That really kind of comes back to focusing on things like fiber, healthy diet, but also tissue repair. That's something that's not focused on so much yet in our field, but there's a really growing amount of research here that specific microbes and their products promote tissue repair. So that's really, when we're talking about gut healing, that's kind of the central part of gut healing. So it's more than just leaky gut. We want to also make sure that tissue repair is promoted. All right, so coming back to the results for this patient that we're focusing on, again, we can see that there's a lot of deficiencies here overall, and that when you see this type of pattern, typically you'll see that that is often linked to a low secretory IgA. We will start diving into some of the specific microbes here and how they support a healthy barrier. But first, I want to just kind of point out that link here. So we did see that the secretory IgA was quite low. And again, you will commonly see that. So this is telling us that the best way to increase secretory IgA is to work on increasing the normal bacteria. Traditionally, there's a lot of supplements, which are certainly very helpful. Saccharomyces boulardii, glutamine, again, zinc carnosine, immunoglobulins, such as colostrum or serum bovine immunoglobulin, and even vitamin A. Those are kind of the top supplements that are known to help promote secretory IgA. But in addition to that, again, we want to consider supporting the normal microbes. And we'll talk about some ways to do that in a little bit. So again, just want to reinforce this link here that when it comes to the secretory IgA, when you see that low, you should be thinking that probably the short-chain fatty acids and the microbes that produce them are low. So we want to focus on, of course, diet and other factors to try to get the secretory IgA to increase. I really like this visual. So it starts to break down where some of these key microbes live. And this is a depiction of the colon, the mucosal barrier in the colon. So you can see that there's, in the outer mucus layer, there's two mucus layers, outer and inner. In the outer mucus layer, we have Akkermansia and several of the key Bacteroides species. So that's why when you see low Akkermansia and low Bacteroides phylum, that can be a key sign that the mucus layer is deficient, since they really thrive on adequate mucus production. But the proteobacteria, so the healthy ones under normal conditions, the main one would be Escherichia. So that's in the commensal section, that's E. coli. So that's the E. E in E. coli stands for Escherichia. We'll look at that in a moment, but that's in the inner mucus layer. Now, we think of E. coli as kind of being something that's bad, especially the pathogens. We don't want to see that elevated because that's associated with inflammation. But it turns out there's a fair amount of research now that Escherichia also promotes, at normal levels, in a healthy gut, it promotes the regeneration of the intestinal lining and mucus. So all of these are important for assessing mucus and just the general health of the mucus barrier. Just to touch on a little bit of the research supporting Akkermansia, I think we're all pretty familiar with Akkermansia. It seems to be pretty famous these days. It's even available as a supplement now, probiotic. It's known to colonize the mucosal layer, as they say here, of the human intestine, where it triggers both host metabolic and immune responses. Akkermansia mucinophila is particularly effective in increasing mucus thickness and increasing gut barrier function. There's even research now that shows that Akkermansia promotes the development of goblet cells that produce mucus. So a really important microbe, not only that it depends on mucus, but it also helps promote mucus production. So the ones that we talked about were the Bacteroides. Many of those live in the mucus layer. We won't get into details there, but some studies indicate that they produce factors that promote a healthy barrier overall, but also healthy mucus. Akkermansia is another one, and then Escherichia. So you'll see in some patients that they don't have such a broad deficiency like we see here. They might just have a few. Sometimes it's just Akkermansia. Sometimes it's just Bacteroides. But in many patients, I'll see all three of these deficient. So that's likely telling us that their colon, especially their colon mucus layer, probably not very healthy. They may need some targeted support. Just also, this is a big topic, which we won't get into. I just want to touch on it here that, again, just reinforcing that the normal gut microbes, the commensals, help protect against both pathogen infections and also pathobionts, which is just another word for opportunists. So they help prevent opportunistic overgrowth. So very important, again, for keeping everything balanced, trying to keep those pathogens away. If you do see pathogens on the first page, even if they're not causing symptoms, so some of those pathogens may be asymptomatic in certain patients, probably it's still telling us that there are deficiencies, such as lack of normal bacteria, low secretory IgA, even low stomach acid, things like that. Just a little bit more about this topic. So this is just one of the key articles, key research studies that came out a few years ago, linking lack of butyrate-producing bacteria to increased growth of pathogens. In this case, they looked at how reduced butyrate producers or reduction in butyrate producers led to an increase in infection with Salmonella. There's many examples now of this process. Of course, they also trace that back to lack of dietary fiber as being one of the factors, but that leads to reduced mucus. And then that, of course, can enhance pathogen susceptibility. So again, really connecting diet and healthy lifestyle with increased susceptibility when patients are not consuming healthy foods. This is depicted here in this visual. So you can see on the left, in a healthy gut, there's plenty of these fiber-degrading microbes. Those are in the green. Here you can see at the bottom, based on the key. And that whole process where they're releasing short-chain fatty acids results in a nice thick mucus layer. So that's a healthy gut. The pathogens that we might be exposed to just don't have easy access to infect the epithelial barrier. But without enough fiber, then we have a lack of these normal butyrate-producing bacteria. The mucus layer is overly degraded, and then these pathogens can multiply. They don't have as much competition, and then they can basically attach to the barrier, produce toxins, they can invade the barrier, and then cause infections. So a really nice visual just kind of illustrating how important that is to make sure that we're supporting the beneficial bacteria. So this is really kind of there's a lot of ways to do this, of course. Again, that could be a whole webinar on just how to support beneficial bacteria, but we summarize this kind of as an easy way to remember with the 4 Ps. So that includes prebiotics, so of course fiber, either supplements or diet or both, of course. Polyphenols, they act synergistically with the prebiotics to help promote healthy beneficial bacteria. So a lot of studies showing that polyphenols not only just support beneficial bacteria, but they also inhibit the bad guys, the opportunists. I think, of course, all of us are familiar with probiotics and their potential beneficial effects. And then the newer category called postbiotics. Currently, that's only a few products right now. That's a really active area of research to basically supply as a supplement some of these beneficial products they produce that are deficient for some people. And that would include butyrate as particularly the main one. And there are a few others available now. So again, really comes back to assessing the barrier, looking at these factors that promote a barrier and are they deficient, but also factors that disrupt the barrier, damage the barrier. This is a good review article from just a few years ago, 2018, called Mechanisms Regulating Intestinal Barrier Integrity and Its Pathological Implications. It has a really nice figure here that summarizes not all of them, but many of the key factors that can disrupt the barrier and how they interact. So I'm going to zoom in here just a little bit and highlight, of course, there's genetic susceptibility, environmental stresses, bad diets, processed foods, various drugs, antibiotics obviously can disrupt the barrier. Some of them alter the normal microbiome, and that's how we end up with damage to the epithelial barrier. Some of them can directly damage the barrier, such as the pathogens, some of the drugs, etc. So just good to keep all these things in mind when you're kind of going through your checklist of things that can disrupt the barrier. Of course, we're going to focus mostly on microbes on GMAP, so the pathogens and opportunists that can disrupt the barrier. So that's these 2 categories. And then also, since they can affect the immune system, they can cause inflammation, we want to focus on that a little bit as well and talk about a couple of those markers. Again, so this patient did not have any pathogens. Those are located on page 1. So these are the most common bacterial pathogens, parasitic pathogens, viral pathogens. Lots and lots of research showing that virtually all the pathogens can disrupt the barrier in different ways. We'll just go through a couple examples here. So this is E. coli, the pathogenic E. coli. So the ones that are on page 1, such as enterohemorrhagic E. coli, enterotoxigenic E. coli. So they say here pathogenic E. coli strains may impair the intestinal barrier in at least 3 different ways. They go into some details here. I won't read through that. I believe you'll have access to the recording so that you'll be able to come back and sort of get some of these details. But the bottom line is that we know that they can disrupt the barrier in various ways. Definitely this is true for C. difficile, which has actually recently been renamed. So it's no longer Clostridium difficile, but they decided to come up with a more complicated name, and I think it's called Clostridioides difficile. So not as easy to say. But we know that that produces a couple of key virulence factors, and that's highlighted here, that C. diff produces toxins that primarily target the intestinal epithelial cells. So they're targeting the barrier for disruption. That's basically what C. diff wants to do. Following toxin endocytosis, so the epithelial cells take up these toxins, toxin A and B, and then they get activated inside the cell. It says the epithelial cells undergo necrosis. That's a type of cell death, which leads to loss of the intestinal membrane integrity, and then host exposure to all of the intestinal microorganisms, which activates the inflammatory response. So it's not just leaky gut, that's part of the picture, but it can actually destroy the epithelial cells. So you really just don't have a barrier at all. So it's not just a little bit leaky. That's depicted here visually from that same review. So you can see here C. diff depicted, and then this TCDA, TCDB, those are toxins A and B. Once they are taken up by these epithelial cells, they basically cause death and disruption of those cells. And then these microbes can get through, stimulate inflammation. So different pathogens can disrupt the barrier in different ways. Candida, definitely well documented to disrupt the barrier. We won't get into details here, but it actually can do that in many different ways. Usually when it transforms from the relatively harmless yeast form. So it can exist in the gut and not necessarily cause a lot of problems, but especially under inflammatory conditions or when there's dysbiosis, then it can form that hyphal, the hyphae, so a filamentous form that extends these projections through the epithelium, and it can disrupt the barrier through that mechanism. It also produces a virulence factor that can disrupt the barrier as well. So Candida, generally, when you see that elevated, it's more likely that it could be causing significant leaky gut. Giardia as well. That's one of its main ways in which it causes problems in the small intestine. Produces virulence factors. It just mentions in the title here, these are called cysteine proteases. Those disrupt the intestinal epithelial tight junctions, and then have other effects as well. So pathogens can do all kinds of damage to the barrier, unfortunately. So what about Pseudomonas? So this patient actually had high Pseudomonas. That was its main, this patient's, this test showed kind of the main imbalance. So this patient had pretty significant levels here, not significant other opportunists, just the Pseudomonas. The patient did have, just to remind you, the patient did have diarrhea dominant IBS. And Pseudomonas is one of the organisms. There's a growing list of different organisms that have been linked to IBS. This is going back more than 10 years. This was published 2011. Several studies around that time linked increased Pseudomonas to IBS. Now we have more idea of why that's the case, but for a while there we only knew about the association. This is one of the potential links. So this is a whole other topic, and we do have other webinars discussing the role of mast cells in IBS and how mast cells can cause abdominal pain, it can disrupt the barrier, etc. This is a nice review article that summarizes a lot of these different microbes that can stimulate mast cells. So certainly Staph aureus, that's probably one of the best documented. Streptococcus, but Pseudomonas is one of them. So that can be one way in which Pseudomonas contributes to IBS symptoms and to barrier dysfunction. We'll look at the details for mast cells in a little bit here, but again, this patient did have increased Pseudomonas. We do know Pseudomonas, one of its effects is to increase mast cell activation. So this is a nice visual kind of summarizing these key effects of mast cell activation in the gut, primarily linked to functional GI disorders like IBS, functional dyspepsia, and potentially some other conditions, food sensitivities, food allergies. So you can see here that various factors can stimulate or activate mast cells, particularly stress, that's well documented, different food components, bacterial products, and also products from the immune system when there's inflammation. That causes the mast cells to degranulate. So basically that's the activation process. Mast cell activation releases all these different components like proteases, histamine, serotonin, and immune factors. And then on the right, we see all these different issues that can cause. So for IBS, that's visceral hypersensitivity. It can cause altered motility. Of course, we're focused on intestinal barrier issues and increased permeability. That is one of the main negative effects of mast cell activation in the gut that can cause leaky gut and potentially even some epithelial damage. And then that also can further activate the immune system as well. We also know that histamine can be produced by a number of microbes in the gut. Some of the best documented ones is Morganella and Klebsiella pneumoniae. So histamine has a two-way interaction with mast cells. Of course, we know mast cells produce histamine, but at the same time, research shows histamine can activate mast cells. So these really interesting study came out last year that was considered a major advance in research on IBS, showing that different Klebsiella species can produce high amounts of histamine that activate mast cells in the gut, particularly in the colon, but probably also in the small intestine, and then cause IBS symptoms, especially abdominal pain. So we know that leaky gut is a common feature of IBS too. So likely there's an issue there as well. With GI map, we have a list here of some of the ones that are best studied as far as promoting leaky gut, or I should say actually histamine. So lactobacillus is the exception here. I only list it because it is known that some can produce histamine. So that may be why some patients don't respond well to probiotics. But generally, lactobacillus is the exception because it's thought to promote a healthy barrier for most people. But some strains might produce a lot of histamine. So just something to be aware of. But by and large, most of these are types of opportunists that we don't want to have. And they can produce LPS, which can promote inflammation. They can promote histamine production as well. So we're almost done with the complicated figures here, but I like the visuals. A lot of people are kind of visually oriented, but it kind of ties everything together here. So I just want to emphasize this. This is from a review article that just was published recently. You can see the reference at the bottom, 2023, so it's brand new. The figure title says Immune Activation in Irritable Bowel Syndrome and Functional Dyspepsia. So we can see here at the top that when you have the combination of dysbiosis, so the microbes and microbial products, and also potentially food antigens, in combination with leaky gut, so we have the impaired barrier here. Those microbes, those microbial products, and the reactions to foods, food sensitivities, etc., those alone can impair the barrier. And then that can result in activation. This is a mast cell here, activation of mast cells. Notice the arrow between impaired barrier and mast cells goes in both directions, two directions. So we know that these issues, dysbiosis, leaky gut, can activate mast cells, and then that can further impair the barrier. So it becomes a little bit of this vicious cycle. Mast cells often work together with eosinophils. So the two tend to sort of activate each other. That's an eosinophil here. Eosinophils also can produce products, which we'll talk about, the one that's on GI map, that can also disrupt the barrier. So that's the connection here between that new marker that we have and the potentially disrupted barrier. But just kind of, again, ties all these pieces together. Microbes that end up activating mast cells can be a problem for the barrier. And also, when you're looking at evidence on stool testing, eosinophil marker also can indicate barrier disruption. So on GI map, that's the eosinophil activation protein. That's just sort of a generic name for this particular factor that actually goes by several names. So you'll see below eosinophil activation protein, it says EDN or EPX. So if you're looking at research, other tests, you might see these other names for it. we wanted it to be more intuitive, so we just kind of gave it a generic name, even though the technical names are below. So we know that that is a sign that eosinophils are activated when it's high. And that's basically this. It's the EDN here, not the MVP. That's another product produced by eosinophils. I just wanted to kind of make that connection for you there, that we know that that can disrupt the barrier. We do see a high eosinophil activation protein in patients that have food sensitivities, food allergies, some IBS patients, not all, and a lot in inflammatory bowel disease patients. That makes sense because they usually have a very disrupted barrier in IBD. So that's when you start to have broader inflammatory involvement, including eosinophils. Of course, there's always calprotectin. You certainly know if that was high. It's not high for this patient, but if it was, that can be another contributor to intestinal barrier dysfunction. Okay, so we've gone through a lot of information here that supports this expanded view of the intestinal barrier. We talked about the microbiome, secreted factors such as mucus, secretory IgA, a little bit about the epithelial layer, especially zonulin, certainly how pathogens can damage the barrier, and then also the mucosal immune system, mast cells, calprotectin, those factors too. So you're really kind of taking this whole picture into account when you're deciding and assessing whether a patient has intestinal barrier dysfunction. Now I want to zoom in a little bit more. This is kind of the last research piece here. This is kind of a newer perspective coming out of research that's not talked about much yet in functional medicine. So on the bad side, we do know that, as I talked about, various pathogens that cause enteric infections can disrupt the barrier. They can also do that by interfering with the gut repair process, and that's highlighted here. So it says, under normal circumstances, gut damage is rapidly repaired by intestinal stem cells. However, with substantial and deep perturbation to the gut lining and systemic dissemination of gut contents, we know that some enteric infections can cause the impairment of this regenerative process. So that's the stem cells. So we're going to talk about that and how normal microbes actually promote this regenerative process and promote stem cell function. That's illustrated here on this figure from a review article. You can see at the top that basically initially when there's an infection, you get inflammation, you get an increase in the dysbiotic microbes, and that can damage epithelial cells. So we've already talked about that. But once you have inflammation, that's going to happen just routinely. If you have a healthy gut, if you have resilience, then this resolution and repair process kicks in and the gut heals. That doesn't always happen, unfortunately. Part of that is because patients may lack these pro-repair microbes. So that's again where the lack of normal microbes can be a problem. Akkermansia turns out to be really especially important for this. This is titled the microenvironment of, this was done in a mouse model, so microenvironment of injured mouse gut, referred to as marine gut, elicits a local pro-restitutive or pro-regeneration microbiota. So they say these data confirm that Akkermansia and other wound-associated bacteria promote mucosal wound repair. And they go through this mechanism here that results in promoting epithelial proliferation. So that's really the whole repair. That's kind of the real meaning of the word gut repair. Certainly leaky gut and closing those tight junctions is part of it, but ultimately your cells are turning over every 3 to 5 days. That's really gut repair. And you want to make sure that process is healthy and happening. I won't go into detail here, but just the title says mucin degrader, Akkermansia accelerates intestinal stem cell mediated epithelial development. So growing amount of evidence that Akkermansia is very important for this aspect of gut healing. Of course, there's Akkermansia. So again, be thinking gut healing. Beyond, again, glutamine and carnosine in the supplements, we also want to make sure the microbes that promote repair are healthy as well. Turns out lactobacillus type species also promote gut healing, as long as it's not a strain that is producing too much histamine, for example. So it's a little bit complicated there, but generally lactobacillus is beneficial. Lactobacillus produces lactate or lactic acid, which accelerates intestinal stem cell development, similar to Akkermansia. So again, you want to see that one at healthy levels. Lactobacillus reuteri also maintains healthy epithelial regeneration. Luckily, there's a lot of probiotics out there now that contain various strains of Lactobacillus reuteri. So that's a good one to look for in formulations. Again, we're looking at the genus level here, not specific species, but in general, when you see it's low, that can be yet another contributor to lack of gut healing. Then we'll wrap up pretty quickly here because we're almost at the top of the hour and we'll take questions. But so it turns out biotin, so there's various supplements, of course. Biotin also plays a role in this repair process, but some of the microbes also produce biotin right there where it's needed. So they talk here below. It says biotin produced by E. coli regulates ISC's intestinal stem cell proliferation. And they talk about Bacteroidetes, Fusobacterium, which can have some downsides. So we'll kind of not talk about that right now because that's a little complicated with Fuso. But then E. coli. So those are two of the key species that we saw in that figure that live in the mucus layer, really close to the epithelium. So one of the things they produce is biotin that helps promote repair. So again, when you see those low, especially Bacteroidetes and E. coli, that can be a sign that the gut healing process needs some support. And again, those are listed here. Oops, go back to that. So Escherichia and Bacteroidetes. We also know that not only microbial metabolites, but also certain dietary components. This is just such a kind of a hot topic in research because in certain conditions where it's difficult to get the gut to heal, particularly in IBD, inflammatory bowel disease, they're looking at this research to see are there other ways besides the standard pharmaceuticals that suppress the immune system, are there ways to promote gut healing? That's the title here, Mechanisms of Mucosal Healing, Treating IBD Without Immunosuppression. And then one of the sections they focus on are these microbial and nutrient-derived metabolites that can promote repair. As far as lifestyle factors, I'm just going to mention a few things here before we wrap up. That fasting is now actually well documented to help improve the health of these intestinal stem cells. So this sort of intermittent fasting, the idea of fasting and then reintroducing food, is one way to help potentially promote repair. These NAD precursor supplements like nicotinamide riboside, we know that intestinal repair goes down during aging and age-related diseases. So this study is one of kind of a growing number of studies showing that these types of kind of healthy aging supplements, you think of them promoting healthy aging. One of the ways in which they can do that is by promoting this stem cell repair in the gut. All right, so that again really comes down to just assessing these factors. What are the factors that promote a healthy barrier? Now you know a lot more about these different microbes in particular and how they promote healthy barrier in different ways, and also the bad guys and how they disrupt the barrier. And we talked about these main components, looking beyond leaky gut. So just quickly now, with all that information behind us, let's go back, just quickly review this patient's, patient 2. Let me actually go back and, so this, just a reminder, this patient had IBSD, gastritis, food sensitivities, mast cell and histamine symptoms, long COVID, and a history of SIBO. So no pathogens. H. pylori looked good. Again, we're back to this screenshot that we've seen a lot of here. This patient had overall very, very significant lack of normal bacteria. So that's probably their number one issue here. So a lot of support for these different types of microbes would likely be helpful. Also dealing with Pseudomonas. Luckily, Pseudomonas actually responds well to the typical broad spectrum antimicrobial herbs. So things like berberine, garlic, oregano, combination formulas that include multiple factors tend to work quite well against Pseudomonas. One other thing to note about Pseudomonas is poor digestion is another factor that can lead to increased Pseudomonas, especially low stomach acid, even low elastase. Lastly would be foods. We know that gluten, dairy, and egg protein in particular can promote the growth of Pseudomonas. In turn, we know Pseudomonas can contribute to food sensitivities. And that could be one reason why those are common foods that cause reactions because of microbes like Pseudomonas that can make those reactions more likely. Again, they didn't have any of these inflammatory microbes, just the Pseudomonas, no candida, no viruses, no parasites. We did see some evidence of mildly reduced digestion, so that could be part of the problem. And then downstream, we have that lack of normal microbes leading to low secretory IgA. So fairly simple scenario. We see a lot of patients that are more complicated, but we can be pretty targeted with this patient because we can see pretty clearly where their intestinal barrier issues are. So that's all the information I have. It's a lot of information, so I imagine you may have some questions. But thank you for listening. I hope you found this information helpful for working with GI map in your clinical practice.
Absolutely. And I like the way that you use cases. It just makes it more like our real world when we are sitting with patients, having to go through the results with our patients ourselves. So it just makes it less scientific and more clinical. Thank you very much for doing it that way. Yes, questions did come up. And there was, let me just see the first one. Yeah, like you already touched a little bit on that one. One was like how, if you give just like a zinc carnosine, or it could be other things to heal the gut with for 12 weeks. But the question was specifically zinc carnosine for 12 weeks. Will that heal the gut? Just that alone? Before you answer, I can give an example of a patient that I had who matched clinically to a leaky gut patient, multiple food intolerances, malnourished, like he looked nearly dead. His skin was like a wax, and the first thing I started giving him was only zinc carnosine as the only thing in small levels. And even the next time when I had to give him results back on stool tests, etc., things had already changed. So, I mean, we can be lucky sometimes to hit the right nail when we choose supplements. So, but you, of course, may have some more experience using some of the zinc carnosine or the glutamine or N-acetylglucosamine, and then if you've seen post results, how quickly does it recover, or how quickly, I mean, when there's lots of dysbiosis and overgrowth of other things and stuff lacking and so on, I guess it won't just do the trick. You need to fix different areas as well.
Yeah, I mean, I can't say that I've seen a lot of specific cases where they used zinc carnosine only and then did a retest. Actually, in fact, I don't think I've seen that particular scenario where they just tried the one thing. Usually it's a full gut healing protocol. And oftentimes it's combined with, if there's opportunists, then they're doing antimicrobials. If there's lack of normal bacteria, then they're doing those 4 Ps that we talked about. So that's a little bit difficult to say based on these retest cases that I come across, whether which of those components works the most for a given patient. But that said, I would say that a lot of practitioners do prefer to kind of start simple with the kind of tried and true approach, see if that is sufficient, if that works, so they're not kind of inundating patients with too many supplements, a difficult protocol. So that's where this information can be helpful when that doesn't work, because then you're kind of widening your scope and looking at what are some of these other factors that were imbalanced on their GI map, and maybe we need to look at doing something different. So I definitely think zinc carnosine is quite helpful, and glutamine as well. They're mostly targeted to the small intestine, so it sort of depends on where their issue is. You can have leaky gut and issues with the barrier in the colon, or the small intestine, or both. So small intestine is the small intestinal cells depend a lot on glutamine. That's one of their main fuel sources in the small intestine. Zinc is a key factor for many of the processes in the small intestine, because we know it's helpful for promoting stomach acid production. It's helpful for, we didn't talk about this at all because this is just kind of beyond the scope, but there's a really important enzyme, brush border enzyme, produced in the small intestine called intestinal alkaline phosphatase. It plays a really large role in maintaining the intestinal barrier in the small intestine. One of the cofactors, from what I remember, is zinc. So that can be one of the ways in which zinc is helpful in the small intestine. In the large intestine, it really does come down to short chain fatty acids, butyrate. The main fuel source for a colon cell, the colon epithelium, is butyrate, and to some extent propionate, so some of those other short chain fatty acids. So that's kind of where, looking at GI map, again, this was sort of beyond the scope today, but we can get a sense to some extent of whether the dysbiosis is happening in the large intestine, that's mostly page 2, or the small intestine. So a lot of those opportunists on page 2 are much more dominant in the small intestine, even just in a normal small intestine, and they can increase when there's dysfunction. So Streptococcus, Staphylococcus, Pseudomonas, for sure, that's been shown to be pretty much just in the small intestine. So this is where you can get a little bit more granular. So if you're trying glutamine, zinc carnosine, and that works, that probably means they had some small intestinal issues. If it doesn't work, and you see low normal bacteria, for example, some of the pathogens, some pathogens are more in the small intestine, some are more in the large intestine. So you can kind of get an idea of where the problems are based on where these microbes live. So just to give a quick example, like C. diff, that one is primarily in the colon. Enterohemorrhagic E. coli is in the colon. Enterotoxigenic E. coli mostly causes problems in the small intestine. Giardia is more small intestine. Candida is more small intestine. So I won't get into too much detail here, but you can start to kind of parse out a little bit and see where you need to support based on the overall picture. And that is more of an advanced, through level of looking at GI map.
Cool. So I was just going to mention some of the supplements. So I was going to mention collagen and nucleotides. Those are some of the things that we have in the system. There's biotin. There's products that is like GI Revive, that has a mix of different leaky gut fixers with some N-acetyl-D-glucosamine and some glutamine and maybe some licorice and other stuff like that. Then there's also something called mega mucosa that has some amino acids in that has some effect on the healing of the leaky gut. And then, of course, the indocene, and then butyrate sometimes the product is called butyric acid, sometimes it's called butyrene or just butyrate. So that the products are there. So then there was a question about if low secretory IgA can be used as a possible indicator of false negative food intolerances. But I guess it depends on how you measure the food intolerances, because if they're IgG, you can't really use a low IgA for food intolerance, but you can also measure food intolerance that is IgA related. So if the IgA, secretory IgA is low, can you perhaps read the gluten, the gliadin IgA, that if the secretory IgA was normal, then maybe the gluten would be higher, or other IgA food intolerances would be higher, or...
Right. Yeah. So it's not 100% well studied. And keep in mind that on GMAP, the secretory IgA is measured differently from the anti-gliadin. They have different units, so you can't compare them sort of directly and say that, if secretory IgA is low and the anti-gliadin is at 150, that, does that mean that anti-gliadin is really actually more like 300 or something? So there is some effect. So the short answer is yes, but only a small effect. If you see high anti-gliadin, those are the direct levels that we're measuring. So it's not a sort of relative measure, it's an absolute measure. So regardless of whether the total secretory IgA is high or low, the amount of anti-gliadin that we measure is that amount. That's the absolute amount. Now, in certain circumstances, if patients have like a genetic deficiency in the ability to produce secretory IgA, they might be reacting to gluten, and you won't see a very high anti-gliadin because they just can't produce, no matter what, they can't produce very much. So you might get some false negatives in those cases, but that's not that common. So to my knowledge, it's not as big of an effect as you might think, more of a minor effect.
Okay. Then the question probably it comes up depending on which test, of course. So which is the best stool test? Which is the best organic acid test? Which is the best way to measure zonulin, blood, or zonulin-like proteins, or zonulin in stool? We just need to have that answer from you as part of this webinar.
Yeah, so there's more research on serum zonulin overall. The concern there with serum zonulin is the source of zonulin is not just the gut. There's other barriers, like the blood-brain barrier, other barriers where zonulin may be produced. So it's not always specific to the gut, whereas the fecal zonulin is obviously going to be specific to the gut. So it's thought that there's some advantages to measuring it directly in the stool. Plus, you're measuring it, closer to the source, so you might miss it when you're doing a serum test. It might look relatively normal, but if you were to do a fecal zonulin, then that might show an elevation. Then there's the second sort of question or issue around zonulin versus sort of the zonulin family peptides. From my knowledge, virtually all companies use the same kit or kits source of what's being measured in that particular assay. And so now they think it may be not necessarily zonulin itself, but something that's called zonulin family peptide. The implications are still the exact same because all of the research has also been done on using that same kit, basically. One key source, I don't remember the company's name, but I believe it's out of Germany, is kind of far and away the most common kit that's used in research in clinical labs. So there's still this strong association with leaky gut, but exactly what's being measured, I've only seen a couple studies that sort of called into question whether it was zonulin or zonulin family. I don't know if that's been fully resolved yet. And I think, so I don't work in the lab for DSL, so I don't know exactly, all of the details, but my understanding is that until definitive research says, yes, this is zonulin family versus just zonulin, that the label we use is zonulin. So it's still kind of that same measure based on all the research on that marker. So it's a little bit of a complicated answer, but that's the best of my knowledge.
That's okay. So then I think the last question, there's other questions, but I'm just going to do the last one because now it's 4:15. So just to round up, or sum up, so if a patient is going to do a GI map, how do they prepare themselves? If they've had antibiotics, how long time should it be? Are they taking probiotics? Are they doing digestive enzymes, etc.? What's your recommendations on that?
We don't have very many sort of limitations other than antibiotics and antimicrobials that can basically disrupt the microbiome. You want to wait a minimum of 30 days. We recommend 30 to 60 days after the end of the protocol so that the microbiome has a chance to re-equilibrate after the treatment. But also, if you're targeting some sort of pathogen or opportunist, if you were to just run a GI map right at the end of your protocol, it might look successful. But we do know that pathogens sometimes can drop below detection. And if your treatment was not successful, then they will grow back. So 30 to 60 days will give you some time to see if these bad microbes are growing back after your treatment. So it just allows you more time to see whether ultimately it's successful or not. As far as any sort of diet or supplements, that's really practitioner specific in terms of, do you want to look at the patient on their current, so what their GI map looks like on their current protocol, their current regimen. There's no contraindications for probiotics or anything else. For lactobacillus and bifidobacterium, the supplements don't seem to have that much of an effect on the levels. It's a little bit different for the spore-based probiotics because normal levels of bacillus in the gut, that's on page 3, the opportunist, it's really more of an overgrowth bacteria. It's not really a bad guy, but it can overgrow when digestion is not good. When that one is, so it's kind of present at a low level. When you add a high dose spore-based probiotic, you can see those numbers go up. So it's just more being aware of that probiotics might affect those particular species, but generally we don't have any contraindications or caveats against any of the diet or supplement regimens. It really depends on, as a practitioner, what do you want to see? You want to see how their gut looks without the treatment or before the treatment, or do you want to see how the treatment is affecting the gut?
Super. One just asked if someone has had a stomach infection or flu, how long time should you wait after you feel you've, recovered? I don't know if you
That's a really good question. So again, we would say 30 to 60 days if possible, but that's going to be highly variable. I mean, I've seen some cases where patients had a gastroenteritis many months before they did the test, and their microbiome still seemed to be disrupted. So some patients recover slowly, some patients recover quickly. So there's no kind of magic number, but in general, we would say wait 30 to 60 days for anything that's going to have a big disruptive impact, where that's not their normal scenario.
I think that's it for now. And thank you very much for your, as always, excellent presentation. And people stayed unhooked for a long time. So they were tied to their computers, learning all your little clinical pearls here. And I, of course, will for sure invite you back. Then I hope you will accept the invitation.
Absolutely. Yeah.
Thank
I
you.
really enjoy interacting with this group. So I really appreciate everyone attending.
Yeah. So, and then, of
Good
course,
question.
everybody else, we will send out the recordings so you have those and the slides. Tom, please share those with me so I can share those with everybody else. And then, yeah, until next time, have a lovely time. And again, thank you, Tom.
All right. Thanks again, everyone. Have a great day.
Linnea, if you have challenges, contact or getting into the portal, please just reach out anywhere to support send an email to info@nordic-labs.com, or just give us a call. And the other one, if you're still there, who was it who asked about refunds? I just wanted to write as well, if a product has been opened, we do not refund it. And if a product is about to expire or close to expiry date, we may not refund it. And if it's probiotics or stuff that needs to be kept in the fridge, or like fish oils where we don't know if it's been stored in the sun or something, then we may not take it back either. But please reach out, and then we can evaluate the specific order. So yeah, I think that was it. Thank you. And I will send out the recording to those who missed it and logged in at the end. Bye.
If you do not already have an account with us, please contact our team and we will assist you. If you found this webinar helpful, please like and subscribe to our channel.